Dr. Narda here, back with another video. And in this video, I would like to talk about the body's senses. In particular, we are going to talk about the somasensory systems, which include proprioception and skin senses. We will also talk a little bit about interoceptive systems and your vestibular system. I know vision and hearing get all the glory, but we have these other important systems as well. So please stay tuned. And if you have not done so already, please like, subscribe, and share. Let's get into this. So the first term I want to define is proprioception. Now, if we're thinking about proprioception, we kind of have to define the term. And this is really the sense that informs us about the position and the movement of our limbs, our body, and our head. We have sensors that report tension and length in muscles, as well as joint angles. We also have senses that track the head as we move about in our environment. And this system is key to many basic functions that we take for granted, such as maintaining posture, moving limbs, grasping objects, and locating placed objects without looking at them, such as grabbing your cell phone out of your pocket or grabbing a fork that is sitting next to you at dinner. It is all about our proprioception systems. And if we did not have our proprioception systems, well, there could be some issues. We'd have floppy, ragdoll-like movement. We would need our visual senses to sit, to stand upright, and to walk. We'd also totally collapse in a dark condition where we can't use our vision to tell us where our body is. Now I want to talk about our skin senses. This is a group of senses that commonly include touch, warmth, cold, texture, and pain. Now, newer research actually identifies tickling and itching, which apparently share their own receptors and neural pathways. And they actually have their own receptor proteins as well. Now, skin senses are distinct, and they have their own receptors and pathways to the brain. These pathways may be called labeled line to the brain. The different types of receptors are free nerve endings. These are simply processes at the end of a neural dendrites that detect warmth, cold, and pain. We also have encapsulated receptors, and these are more complex structures enclosed in a membrane. Their role is actually to detect touch. We even have specialized touch receptors that assist us in detecting texture and fine detail. Our touch receptors are located deeper in the skin and contribute to our perception of the shape of a grasped object by detecting the stretching of the skin. Now, sensitivity of your touch receptors vary depending on the density of the skin receptors. For example, fingertips and lips are the most sensitive because they have the greatest density of receptors. Now, your upper arms and your calves of your legs are least sensitive. They have the lowest receptor density. The other three skin senses are detected by free nerve endings. Those nerve endings have distinctly different receptors that make the neurons stimulus specific. At least two different receptors detect different levels of warmth, while another responds to cold. The temperature receptors are all members of the transient receptor potential or TRP family of protein ion channels. Now, detection of pain uses a variety of receptors to respond to three distinct types of pain. We have thermal receptors. They're called the 2-TRP receptors. They respond to pain, heat, while receptors for painful cold are unknown. This is, by the way, not the same as the coolness receptor for skin temperature. We have receptors that respond to chemical, and these respond to a wide range of chemical irritants. For example, they'll respond to hot peppers, if you ever eat anything really hot, or coolness of mint or menthol. We also have mechanical receptors, and these receptors have not yet been determined, which is unfortunate, as touch hypersensitivity is a common problem after tissue or nerve damage. The next system I want to talk about is our vestibular system. Now, this is a sense that helps us maintain balance and provides information about the head position and movement. This is maintained by fluid-filled vestibular organ of the inner ear. Now, structures of the vestibular organ include 3 semicircle canals, which are arranged in different planes of orientation and help the body respond to head movement. At the base of each canal is a gelatinous, jelly-like mass called the copula, which has a tuft of hair cells protruding into it. During acceleration, which is the increase in the rate of movement, the head and the copula moves, but the fluid does not, which causes the copula to bend in the opposite direction of the head movement proportionally to the rate of acceleration. This displacement bends the hairs attaching to the copula to the semicircular canal, which depolarizes them and increases the neural rate of firing. Moving the head in the opposite direction bends the hairs in the other direction, which hyperpolarizes the neuron and decreases the firing rate. The system responds only to acceleration and stops responding when the speed stabilizes. Just like if you have a cup of coffee in your car and you start driving at a traffic light, the coffee kind of sloshes around in your cup, but after your speed stabilizes, the movement will level off. And this is important, otherwise you would continue feeling the movement in your car the whole time, or you'd feel the movement in a plane going 500 miles per hour. So you might be wondering, what is the function of the saccule and utricle? Well, receptors, hair cells, are, again, as I said, covered with a gelatinous mass embedded with small calcium crystals called otoliths. When the head tilts, gravity shifts the mass of otoliths to depolarize or hyperpolarize the hair cells again, depending on the direction. The brain combines information about the object's spatial location with the inputs from your vestibular senses and from the proprioception to tell us what arm and hand movements are required to reach for an object. Movement encoded by the vestibular nerve is processed by the vestibular nuclei in the midbrain, which then sends information to the ocular nerves to produce compensatory eye movements. This allows us to repeatedly focus our gaze to target objects during movement, preventing moving details from fading into a blur. The semicircular canals and the utricle and succula also send projections to the cerebellum and the brainstem. And there is also a pathway to the cortex, specifically to the area called the PIV, or the parieto-insular vestibular cortex. And by the way, if you're wondering, if you ever read in the car, it's those excessive eye movements that triggers the dizziness and nausea. Now, the body is divided into segments called dermatomes, each served by a spinal nerve. The divisions are not as distinct as the illustration shows on the screen, because each dermatome overlaps the next one by 1/3 to 1/2. This way, if a nerve is injured, the area will not lose all sensation. Body sense information enters the spinal cord via the spinal nerves or the brain via the cranial nerves, crosses over the midline in the medulla, and travels to the thalamus. From there, the body sense neurons go to their projection areas, the somasensory cortex, located in the parietal lobes just behind the primary motor cortex and the central sulcus. As with the auditory system, most of the neurons cross from one side of the body to the other side of the brain. So the touch of an object held in your right hand is registered mostly in the left hemisphere. Because not all neurons cross over, touching the object also stimulates the right somasensory cortex, though to a much less extent. Now, the somasensory cortex is the projection area for the body's sense neurons located in the parietal lobes just behind the primary cortex and the central sulcus, as I previously stated. Another area of the brain you should know is the somatopic map. That's a representation of the body in the somasensory cortex, with adjacent body parts represented in adjacent parts of the cortex. Some cortical cells have complex receptor fields on the skin, including excitatory center and inhibitory surrounding fields like those of the visual system. The primary somasensory cortex is a group of 4 areas, each of which contain a somatotopic map of the body and plays a role in processing sensory information from the body. Information is hierarchically passed from the thalamus to the 2 sub-areas, which extract some information and pass the results on to the other 2 areas. The other 2 sub-areas then process information and pass the results on to the second somasensory cortex. At this point, information from the right and left sides remain mostly segregated. So the secondary somasensory cortex receives information from the left and right primary somasensory cortices and integrates that information. Neurons in this area are particularly responsive to stimuli that have acquired meaning, and this area sends connections to the parts of the temporal lobe that include the hippocampus, which indicates that the hippocampus may be involved in forming somasensory memories. The posterior parietal cortex is an association area that brings together the body's senses, such as vision and hearing. It integrates the body with the world by determining the body's orientation in space. Subareas are responsive to different sense modalities and not solely perceptual. Neurons fire before and during movement to pass on information to the frontal areas that trigger the movement. The last topic I want to cover in this video is how your body detects pain. Pain begins when certain free nerve endings are stimulated by intense pressure or temperature, by tissue damage, or by various chemicals. Now, there's something called an inflammatory soup. This is an array of signaling molecules released when the body is in pain, which includes histamines, proteins. You might have heard of one of substance P, lipids, could be neurotransmitters like serotonin. Some molecules stimulate pain receptors while others trigger swelling or redness. Many enhance excitability of pain neurons so much that the neurons respond to even light touch, which encourages someone to guard the injured area. Large myelinated A delta fibers rapidly transmit sharp, stinging pain upon injury. Smaller C fibers, often with little or no myelination, more slowly transmit dull, aching pain after injury. This makes A delta fibers, which are densely packed and superficially located, a good danger signal. C fibers, which are located deeper under the skin, remind you that you still have been injured. Now, there is something called substance P, and it's a neuropeptide that increases pain sensitivity. Glutamate for mild pain, and eventually substance P for intense pain, are released in the spinal cord and enhance sensitivity. Now, we do have an internal mechanism to deal with pain, which are called endorphins. And these are neurochemicals that function both as neurotransmitters and as hormones. They act as an opiate receptor in many parts of the nervous system. Endorphins only work under highly specific conditions. Pain that is inescapable is more effective in causing endorphin release. Well, that's it for this video. Thank you all for watching and making it to the end of the video. Please like, comment, and share this video. I'll see you all next time. Take care. Bye-bye.